Do it. I'm guessing if I gave everybody who came to class an A today, then they would never come to class again. That's just my hunch, which would kind of be a self-defeating thing, right? So
Not necessarily.
maybe we would, right? Yeah, okay. What you should say is, Ahern, you're a scientist, let's do the experiment and find out, right?
Exactly.
Well, we can't do that. I could give you extra. There's a lot of things I could do. I could give you money, right? We could go have beer. We could have pizza.
Yeah, how many of us would not get in trouble for you buying us?
Some of us are still underage, so.
Yeah.
Well, no, actually the way I do that is I go to a place where they can serve people underage, and then you have to show an ID, so it's not my responsibility then. So, yeah. Okay. Well, a lot of energy. I hope everybody's got a big Thanksgiving planned. Wild plans?
Family.
Oh, family, Like I said, if any of you are in town and would like to come over, give us a holler. I have to let you know where I live and everything. But if you'd like to come over, we have plenty of turkey and other things. And no, I won't get you drunk. But we'll have a good time. Today we're going to have a good time because we're going to be thinking about making of glucose. And I know that for many of you that's been something you've dreamed of doing. And you're going to get that dream today. Happy days are here again. Glucose synthesis is an interesting process. The phenomenon, of course, is known as gluconeogenesis. And it is a pathway that is very similar, excuse me, very similar to glycolysis. Very similar, all right? It's very similar to the reverse of glycolysis. However, there are important differences, and specifically, there are two reactions. I'm sorry, specifically, there are three reactions. I can't remember. There are three reactions in gluconeogenesis that are replaced, in glycolysis, that are replaced by four reactions in gluconeogenesis. So gluconeogenesis has 11 steps, glycolysis had 10. And one of the steps takes 2 steps to get around. So it's a 2-step, all right? If you learned glycolysis, gluconeogenesis, for 8 of the steps, okay, so get that right, for 7 of the steps, I can't even get my head today, for 7 of the steps is identical to glycolysis except for in the reverse. Same enzymes, same as intermediates, going the opposite direction. Okay? For 3 of the steps that are in glycolysis, as they say, they're replaced by 4 steps. So let's take a quick look at that. Before I take a look at that, I'll show you something your book gets distracted by, and that's this process here, which is the making of glucose from glycerol. Why do we care about the making of glucose from glycerol? Well, one of the reasons we care about the making of glucose from glycerol is a byproduct of fat metabolism. And so it turns out that the only portion of the fat molecule that can be converted directly into glucose is the glycerol. We don't convert fat into glucose for the most part, with the exception of the glycerol. So I just show you this. I'm not going to go through and expect you to memorize these or anything, but just show you that glycerol is a 3-carbon molecule. It gets made in a couple of reactions into an intermediate in glycolysis, dihydroxyacetone phosphate. And of course, once it's dihydroxyacetone phosphate, we can now do the upwards pathway going into making glucose via gluconeogenesis. Okay, and we see this. This is a phenomenon you've seen before. We saw how other sugars entered glycolysis and got broken down by being converted into glycolysis intermediates. We saw, for example, fructose got converted to fructose 6-phosphate and then got metabolized as an intermediate in glycolysis. In this case, we see glycerol being converted into an intermediate in glycolysis or gluconeogenesis. It could actually go either way and be made into either glucose or ultimately into pyruvate. Okay, well, let's focus on gluconeogenesis since that's our main topic of the day. You'll notice in looking at this screen that we orient it very much like we oriented glycolysis, except that we're going upwards in gluconeogenesis, whereas we were going downwards in glycolysis. So we start at the bottom, and the place where we will start gluconeogenesis is actually pyruvate. But again, we remember that all of these designations about where something starts and stops is really arbitrary. We could just as easily start it at lactate for some types of metabolism. We could start at amino acids as well, but we're going to start it right here at pyruvate. So starting at pyruvate, and that's a good place to start because that's where we finished glycolysis. Starting with pyruvate, cells can convert pyruvate into glucose. Well, not surprisingly, if pyruvate is a 3-carbon molecule and we want to make a 6-carbon glucose, we need to have 2 pyruvates to start everything. So we're going to start with 2 of everything, and eventually they're going to combine into 1 as we get higher up in the pathway. Now, what we discover in gluconeogenesis is the first instance that we see of a phenomenon I call sequestration. Sequestration, meaning we're sequestering something. All of glycolysis occurs in the cytoplasm of the cell. All of the enzymes of glycolysis are found in the cytoplasm of the cell. Okay, Gesundheit. In the case of gluconeogenesis, we see 2 enzymes that are not found in the cytoplasm of the cell. These are sequestered into other organelles in the cell, and I'll show you those. They actually end up being the first and the last enzyme in the pathway. Okay? All the other enzymes between the first and the last are all found in the cytoplasm. All right. Well, let's look at what's happening in making glucose from pyruvate. If we recall in glycolysis, in going from PEP to pyruvate, I said that was the Big Bang. I said that was a reaction that was extraordinarily energetic. It had a large delta G 0 prime. And as a consequence, that we might imagine going in the reverse direction will be an enormous energy barrier to overcome. And in fact, that's exactly what it is. So it's because of this enormous energy barrier that cells can't go directly back from making pyruvate to PEP in 1 step. They have to do a 2-step around it. And the 2 steps around it are these 2 enzymes here, Pyruvate Carboxylase and Phosphoenol Pyruvate Carboxykinase, which you are more than welcome to abbreviate as PEPCK. Okay? PEPCK. Well, let's talk about the first one first, Pyruvate Carboxylase. Pyruvate Carboxylase is an enzyme that is found in the mitochondrion of cells. It's not found in the cytoplasm. So the very first reaction of gluconeogenesis occurs in the mitochondrion, not in the cytoplasm. Okay? In this reaction, as you can see on the screen, carbon dioxide in the form of bicarbonate and ATP are used to convert pyruvate into oxaloacetate. And you can see the structure here. Here's a 3-carbon, here's a 4-carbon over here. We've put an additional carboxyl group onto the end of pyruvate. The carboxyl group going right here. We can see the new carboxyl group on the right side. Here was what pyruvate looked like, what pyruvate looked like over here. So what we did was we took this methyl group and we added a carboxyl group to it. That takes energy to put that on there. It makes a 4-carbon intermediate, and that 4-carbon intermediate you're going to hear a lot about next term because oxaloacetate is one of those molecules that appears in so many metabolic pathways. It's a very, very important molecule. It's important in amino acid metabolism. It's important in the citric acid cycle. And it's also important, as you can see here, in the synthesis of glucose. This is an energy-requiring reaction, so if we started with 2 molecules of pyruvate, it's going to take 2 molecules of ATP and 2 molecules of bicarbonate to make 2 molecules of oxaloacetate. Okay? Well, we haven't gotten to PEP yet because even with all that energy that we've put in, we've made a 4-carbon intermediate, and we have to convert that 4-carbon intermediate into phosphoenol pyruvate, or PEP. To do that, the oxaloacetate, which was made in the mitochondrion, has to be moved out of the mitochondrion and into the cytoplasm. And next term we'll talk about how molecules move across an organelle, but it turns out that there are specific proteins that will shuttle a molecule across a membrane. So there are specific proteins that will transport oxaloacetate out into the cytoplasm. When it's out in the cytoplasm, oxaloacetate can be acted upon by this second enzyme that's unique, and by the way, all the unique enzymes are shown in red on here, by the unique enzyme phosphoenol pyruvate carboxykinase, or PEPCK. notice that it also takes energy, and that energy in this case comes from GTP. Okay? So GTP is, just like ATP, a high energy source. GTP is used in some places in the cell for energy. The most common place we actually see GTP used is in the synthesis of proteins, because all proteins are made using GTP, not ATP. We'll talk about that next term. All right. Well, if we have 2 molecules of oxaloacetate and we want to make 2 molecules of PEP, it takes us 2 molecules of GTP and this enzyme to accomplish that. In the process, a CO2 is released. So look what we did. We put a CO2 on in the form of bicarbonate, and we released the CO2 up here. So no net gain of carbons have occurred. We've done a molecular rearrangement, and in the process of doing that molecular rearrangement, we've also put a phosphate onto the molecule, creating that very high energy PEP molecule. Okay? Now, as I said, this reaction occurs out in the cytoplasm. Okay? And to go from pyruvate to PEP in terms of synthesizing glucose, we had to use 2 high-energy phosphates for each molecule, or a total of 4. So in order to go from here, 2 pyruvates to 2 PEPs, we needed 4 triphosphates, 2 ATPs and 2 GTPs. From an energy perspective, GTP is exactly equivalent to ATP. There's no difference. Okay? Well, going down, if you recall, when we went from PEP to pyruvate, we only got a total of 1 ATP for each one, or a total of 2 ATPs. So we can see that building molecules in anabolic pathways takes more energy than we get out in catabolic pathways. That's not surprising. Okay? Now, we're thinking, okay, well, we're going, yes?
So how does this prevent the PEP just immediately switching back to pyruvate?
Oh, okay. Her question is, she's reading my mind. So my next point is, her question was, how does the cell keep PEP from just going back to pyruvate, right? Well, that's a very, very important consideration, because we know that if that molecule has the opportunity to go back to pyruvate in the presence of pyruvate kinase, it's going to do it. That's the Big Bang reaction, right? We see now why we have to regulate that pyruvate kinase, because if we want this thing to go upwards, we darn sure don't want to be turning this right back around and making pyruvate, because we will have destroyed our purpose and we will have wasted energy and we would have gotten nowhere. Okay? This business of wasting energy and getting nowhere, where we're making something and breaking it down, going in sort of a circle, is something that's called, that we'll talk about later, but it is a non-productive metabolic process that can occur in cells. We don't want that to happen. So for that reason, we want to turn off pyruvate kinase when we're turning on this process. Similarly, we want to turn these off when we are turning on the pyruvate kinase. Everybody follow? Okay. Well, so can I go through that again? So basically, we want to turn off the enzymes of gluconeogenesis when we have on the enzymes that's catalyzing those big reactions of glycolysis, in this case, pyruvate kinase. Conversely, we want to turn off pyruvate kinase, or turn on pyruvate kinase, and we turn these guys off. So we want to have them on one versus the other. Okay? There's a name for what occurs if we put both of these on at the same time. So let's imagine we had a situation in the cell where these enzymes were active, and so is pyruvate kinase. Okay? The cell would turn pyruvate into PEP, pyruvate kinase would turn it back to pyruvate, and we'd go around and around and around and around. That phenomenon is known as a futile cycle, F-U-T-I-L-E cycle. It's futile because the cell is getting nothing out of it. It's burning 4 triphosphates going up and only getting 2 back on the way down. So each time it turns the cycle, it's losing 2 triphosphates. It's also producing one thing. What's the one thing it's producing? No, there's no net carbon dioxide because it goes in and it goes out. It's heat. Heat, right? Just like we talked about exercising, heat's generated. This process will generate heat, and it's going to be totally wasted. Totally wasted. So we don't want to be running these two processes at the same time. Okay. Well, for the moment, we will say, yes, we've got the pyruvate kinase turned off. So PEP starts to accumulate. When PEP starts to accumulate, now the reverse reaction of glycolysis is favored, catalyzed by enolase, and we convert PEP into 2-phosphoglycerate. Okay? Next, we convert 2-phosphoglycerate into 3-phosphoglycerate. And next, we convert 3-phosphoglycerate into 1,3-bisphosphoglycerate. And we remember now that in going from here upwards, we have to use ATP again. So now we've got to put 2 more ATPs into the process. Okay? We get to 1,3-bisphosphoglycerate, and we want to go back to glyceraldehyde 3-phosphate. Now we have to reduce that molecule. We have to use electrons from NADH to convert the 1,3-bisphosphoglycerate into glyceraldehyde 3-phosphate. That involves loss of a phosphate as well. Now we've got 2 molecules of glyceraldehyde 3-phosphate. Our triose phosphate isomerase converts one of them to DHAP, leaves the other one alone. They combine together to make fructose 1,6-bisphosphate. So we're climbing the ladder. And you see in each case, all that we're doing is we're reversing every blue enzyme reaction. We're just reversing. We're going upwards instead of going downwards. How do we do that? We do it by increasing the concentration of these from the bottom, filling upwards. When we get to fructose 1,6-bisphosphate, we have another consideration. Okay? The other consideration is that if you recall during the discussion of the glycolysis pathway, I said that PEPCK catalyzed a reaction that released a lot of energy. And why did it release a lot of energy? I said if we did the reaction with just phosphate, it wasn't very favorable, but we used something to make this reaction favorable. What was it? ATP. This reaction became energetically favorable in the glycolysis direction going down because we put ATP energy in. One thing that we could do is we could say, okay, well, let's reverse that reaction and we'll remake that ATP. That would be tough for us to do because A, the reaction is very favorable energetically going down. That doesn't make much sense to try to do, so instead we do a different reaction. So instead of trying to remake that ATP, we use a different reaction and we use, consequently, a different enzyme. The enzyme that we use to catalyze the conversion of fructose 1,6-bisphosphate into fructose 6-phosphate is this enzyme known as fructose 1,6-bisphosphatase. Now you start seeing these names start sounding like the intermediates. I'm going to help you on this one. We're going to call this guy FBPase 1. FBPase 1. All right? That name will sound different than fructose 1,6-bisphosphatase. Okay? So we're just going to call the enzyme by a different name. You'll see why later I want to call that enzyme FBPase 1. All right? And now what we're doing is instead of remaking ATP by a reversal of the reaction, we're simply clipping off a phosphate. It turns out that's energetically favorable to clip off a phosphate. Why? Because remember, phosphate bonds are higher energy, and if we just simply clip it off, we make that upwards reaction become favorable. So it's a very cool trick that the cell is doing to make fructose 6-phosphate from fructose 1,6-bisphosphate. Questions about that? Yes, sir?
It's not about this, but is there times when the body chooses to run that futile cycle as a means of heat generation intentionally?
Ooh, very good question. His question is, are there times that the body runs futile cycles to make heat? As a matter of fact, it turns out there are. Not this reaction, but other reactions I'll talk about next term. And one is a very important consideration in something in our body called brown fat. And it is a way of helping to up the temperature. It's not this reaction, but another reaction that's done in a futile sense. But very good question, yeah. Other questions? Okay. So we're getting near the end. We're at fructose 6-phosphate. We need to convert that back to glucose 6-phosphate. That's simply a reversal of the reaction of glycolysis. So again, we use the phosphoglucose isomerase to make that isomerization, and we're at glucose 6-phosphate. At glucose 6-phosphate, we have exactly the same problem that we had with converting fructose 1,6-bisphosphate into fructose 6-phosphate. If we try to simply reverse the glycolysis reaction, we would have to make ATP. That would be an energetically unfavorable reaction. It wouldn't make much sense for us to do. So instead, cells use a different enzyme to catalyze a different reaction. The reaction that they catalyze is, again, parallel to the one catalyzed by FBPase 1, and that is we simply clip the phosphate off of this guy to make free glucose. Now, this last enzyme is found only in the endoplasmic reticulum of cells. It's only found in the endoplasmic reticulum of cells. Okay, now, if we look at this, what we see is, here's the glucose 6-phosphatase. This is what it looks like in the membrane of the endoplasmic reticulum. This glucose 6-phosphatase, you can see, is embedded in the membrane of the endoplasmic reticulum. And in order for glucose 6-phosphate to be converted, it must be moved first into the endoplasmic reticulum. And here's another one of those proteins that does the transport of specific molecules. In this case, it's moving glucose 6-phosphate into the endoplasmic reticulum. There it interacts with the enzyme, gets its phosphate clipped off, and then both of them are kicked out into the cytoplasm. Okay, so as a result of that, cells now have made a functional glucose molecule starting with 2 pyruvates, and they're left with 1 glucose. In the process of making this glucose, they have required 6 triphosphates, 4 ATPs and 2 GTPs. They've also required 2 NADHs, and obviously 2 pyruvates to start the process. It takes more energy to make a glucose than we get out of glucose when we burn it. That's why we have to eat. Because if we relied only on our energy from that which we made and then broke down and made and broken down, we would run out of energy. We have to eat to make up that deficit of energy. Connie?
So just to clarify, you need 2 NADHs, 4 GTPs, 2 ATPs, and 2 pyruvates?
No. You need 2 NADHs, 2 GTPs, 4 ATPs, and 2 pyruvates. Okay? So there's only 2 GTPs. That's the reaction of PEPCK. Okay, so we have made that. Now, I want to tell you a little something about gluconeogenesis that's important, and that is gluconeogenesis is not found in every cell of our body. In fact, it's fairly carefully sequestered, again, as it were, not now where in the cell, but actually where in the body. All right? So the primary organs that we have in our body that make glucose, okay, are our liver, part of our kidney. That's the 2 primary places that we make glucose. So muscle cells, for example, do not make glucose. Muscle cells are really good at burning glucose. They're not designed to make glucose. That means that when we're running and we're exercising very heavily, we have to have a way of getting that glucose that's made in the kidney and, more importantly, in the liver into our muscles. That's why our bloodstream is very important. That's why our heart starts beating faster when we start exercising more heavily, is to carry more nutrients in the form of oxygen, in the form of glucose, and in the form of carrying away carbon dioxide. So all those things are important when we're exercising. That's one of the reasons why our blood flow increases as a result of that. Glucose turns out to be a wonderful compound for this purpose because glucose is very soluble in water. It can move in the bloodstream very easily because it's an aqueous environment. The liver dumps it into the bloodstream and poof, it's off to its target tissues in seconds. It gets there very, very quickly. Okay? So glucose is very, very useful for that. As we will see next term, fat is not so good for that quick energy because A, fat is not water soluble. B, fat has to be packaged up into lipoprotein complexes that have to be made to be able to be soluble in the water aqueous environment. Okay, so that's a broad view of gluconeogenesis. Now, gluconeogenesis, as I said, if you know glycolysis, you basically know gluconeogenesis because gluconeogenesis uses 7 of the enzymes that are the reversal of those of glycolysis. And yes, you should know the 4 enzymes of gluconeogenesis that are different from those other enzymes in glycolysis. Okay? I'm not asking you to know additional structures, though. I'm not asking you to know additional structures. Does glucose 6-phosphatase have an acronym? If you want to call it G6PAs, you may. Okay? The other thing I noticed I didn't mention here, and I'll mention it very briefly, is the enzyme Pyruvate Carboxylase. That was the first enzyme that I talked about. That was found in the mitochondrion. We'll see next term why that's kind of an important thing. But it catalyzes the reaction that you see on the screen. There's the oxaloacetate molecule. There's the ATP, the carbon dioxide, actually in the form of bicarbonate. Carbon dioxide, that's all the same thing. The enzyme is one that uses a coenzyme. I haven't really talked about coenzymes yet, and I want to say just a brief word about them. Coenzymes are molecules that enzymes use to help catalyze a reaction. So they're a non-amino acid that an enzyme uses to help it catalyze a reaction. That's what a coenzyme is. The coenzyme that pyruvate carboxylase uses is one you'll see commonly for reactions that involve addition of a carbon dioxide to something. The coenzyme it uses is known as biotin. And biotin is really great at grabbing onto carbon dioxide and giving it to the enzyme to do something with. That's what biotin does. Whenever you see the name carboxylase in an enzyme name, it tells you, A, that it's putting carbon dioxide onto something, and as a consequence of that, it almost always uses biotin to help it do that. It turns out that the carbon dioxide is carried at this end of the molecule out here. The lysine is the place where it attaches to the protein. Okay? So the protein has a lysine side chain, the biotin gets attached, and out here there's a carbon dioxide that this biotin will carry to the active site of the enzyme so the enzyme can use that carbon dioxide. Okay. And PEPCK, just a brief word about that. There's the reaction that it catalyzes. PEPCK is one of those enzymes, although your book shows some allosteric effectors, it's really not very regulated allosterically. A much more important regulation of this enzyme is control of where it's synthesized. So for example, my muscle cells are not going to make PEPCK because they're not going to go through gluconeogenesis. There's no reason for them to have and use that enzyme. My liver cells, on the other hand, which do go through the reactions of gluconeogenesis, my liver cells, which do have the reactions of gluconeogenesis, will, in fact, make PEPCK. Now, what I've just introduced to you, probably didn't realize, was a third mechanism of controlling enzymes. We talked about 3 earlier in the term. One was allosteric control. Second was covalent modification. And now the third is whether or not an enzyme is made. If the enzyme's not made, that's the ultimate control. Okay? So PEPCK is regulated primarily by whether or not a cell makes it. That, of course, involves control of transcription and or translation, and we'll talk about that next term. Okay, questions about that? You guys look like you're asleep today. Why is everybody smirking? Yes, sir.
So when you eat like a meal that's really full of fat,
Yes?
and then you feel super lethargic,
Yep.
is it just your body's trying to get putting all its energy to breaking it down?
So when you eat a meal that's super high in fat and you feel, how did you say?
Lethargic?
Lethargic. Okay. Is that because your body is putting all of its attention into breaking down that fat? Partly. One of the things that happens with eating a meal of anything, even if it's not a big meal of fat, is that your digestive system diverts a lot of blood supply to it to help carry things away. So instead of more blood being available to your muscles and brain and so on and so forth, your digestive system is kind of taking over. And so as a consequence, there's less oxygen for you to think, and there's less oxygen for your muscles and so forth to do things. So it's a natural response of your body that with that lower supply that you're just not going to feel like going out and doing stuff. Yeah.
Like tomorrow when we all eat.
Tomorrow.
Amounts, is that...
So when we eat, I believe you said ungodly amounts tomorrow. Is that going to be a factor? It is going to be a factor. One of the things that some people say is a factor with Thanksgiving, if you eat a lot of turkey, the claim is that turkey is full of tryptophan, and tryptophan can be converted into molecules that help you to sleep. That's a little controversial, so whether that is true or not, I don't know, but I'm willing to take the risk. So, other questions? Would you guys like to sing a song? All right, let's sing a song. I've got two songs. Maybe we should sing. Let's sing the short one first, okay? To do this one, I got to get you in the right frame of mind, okay? The right frame of mind goes as follows. You know, there was a song that was written back in the 1930s. The 1930s was the Depression, and everybody was very upset. Kind of like you guys will be after the lecture's over today. Oh, damn, I don't get to hear more biochemistry. They're very upset. They're very depressed. And so they needed something to build them up, right? They needed something to make them feel better. And America's songwriters created some really amazing songs of that time, okay? And I'm going to get you started on one of them, okay? And you'll see why I get you started on this song in just a second. The song was called Happy Days Are Here Again. Does anybody know the song? Okay, we're going to start to sing this song together, right? Happy days are here again, the skies above are clear again. Let us sing a song of cheer again. Happy days are here again. Aren't you happier now? Happy days are here again, the skies above are clear again. Let us sing a song of cheer again. Happy days are here again. One more time. Happy days are here again, the skies above are clear again. Let us sing a song of cheer again. Happy days are here again. Next verse. Crappy days are here again. The sky above's not clear again. And the sun has disappeared again. Crappy days are here again. Rain is falling from the sky. I wish I knew the reason why. Guess I'll have to wait until July for the weather to be dry. I do not mean to harangue, since rain provides yin and yang. Because the flowers, everyone, love moisture followed by the sun. Let's stay happy till the rain is done in Corvallis, Oregon. Now don't you feel better? I know you do. Pretty much, yeah. This time of year it's kind of relevant, I think. So, okay. Now, I have another one, but we'll save it until I tell you about a couple other things. And I'm actually going to be easy on you guys today because you came here on a tough day, right? Thanksgiving's tomorrow and everybody else is leaving town, and why didn't he give a pop quiz so that we got extra credit, you know, and why didn't he give us those A's that he talked about and so forth? So I'm not going to go off the deep end. I'm going to save that for Monday. Going off the deep end is something I can do, and I figured, why should we do that now? Let's do it Monday. But I'm going to tell you about a cycle that you'll find very interesting and very easy to understand, okay? It's called the Cori cycle. I'm going to skip this and come back and talk about this on Monday. But the Cori cycle is what I want to finish with, and then we'll sing one more song. So the Cori cycle is an important cycle that was discovered by a husband and wife team named Cori. And it turns out to be of critical importance in our body, okay? The Cori cycle is a way for our body to handle very diverse sets of exercise situations, okay? So let's imagine, and forget the screen for the moment, but let's just imagine that I am out on my morning jog, which I haven't been able to do all week because it's been raining and I've been sick. Okay? But I'm out for my morning jog, which I'll be out tomorrow morning. Anybody wants to run, come with me. Out running, and I will take off from my house and go for a ways. Well, I'll fairly quickly, my body will fairly quickly recognize that my muscles really want glucose, because my muscles burn glucose to get pyruvate, ultimately to get ATP, and all kinds of things from that. My muscles don't have a tremendous amount of glucose in them, so my liver wakes up and starts producing glucose. That epinephrine hormone that we talked about, one of the things that it does is it stimulates the release of glucose by the liver. Okay? So my liver takes that glucose and it dumps it into the bloodstream because the liver doesn't need the glucose. The muscle cells need the glucose. The glucose travels to my muscle cells, it gets to the muscle cells, and the muscle cells go, thank you very much. Yeah?
So it's doing more of releasing previously stored glucose, not running glyconeogenesis.
So our question is it releasing previously stored glucose or doing gluconeogenesis? The answer is it's doing both.
Oh.
Okay? All right. So the liver releases this glucose, the muscle cells grab it, and I keep running and running, and I'm an old guy, so I have a pretty good heart, but my heart probably isn't delivering as much blood and as much oxygen as fast as my muscle cells could use it. That's especially true the longer that I run. So the longer I run, the less oxygen my muscle cells are going to have. My muscle cells are going to take this glucose, and as long as they've got oxygen and they can make pyruvate and acetyl-CoA, they're happy. But what happens when they start running out of oxygen? Well, we remember that the only thing that they can do to keep glycolysis going at that point is convert pyruvate into lactate, and they do that. Well, lactate, as I said in class at the time I mentioned it, is a biological dead end. It doesn't go to anything else. All we can do is convert it back to pyruvate. And to do that, we need oxygen. Well, the muscle cell doesn't have oxygen. Lactate's sitting there. It's not doing the muscle cell any good. Moreover, lactate is an acid, so it's starting to drop the pH of the muscle cell, and that's a problem. Okay? Well, the muscle cell says, hell with that, and it dumps lactate out into the bloodstream. And the bloodstream goes right back to the liver, which has plenty of oxygen because the liver is close to your lungs. It converts that lactate to pyruvate, and then, boingo, it does gluconeogenesis. So what we see is a cycle that's occurring in the body, and it's known as the Cori cycle. The liver is making glucose, dumping it in the bloodstream. The muscles are using the glucose, making ultimately lactate when they run out of oxygen. Lactate's going back in the bloodstream and back up to the liver. It's a beautiful system, and it works amazingly well. Make sense? And it's, one last thing. This cycle is needed because, again, muscle cells can't make their own glucose. They're depending upon the liver. And it makes very good sense for the liver to do it because the liver has plenty of oxygen. The muscle cells don't have plenty of oxygen. Yes, sir?
But eventually the liver will run low on oxygen. I mean, the oxygen demands will eventually outstrip your lung and heart's ability to keep up, right? So that's like hitting the wall in marathon running or something?
So his question is hitting the wall in marathon running the equivalent of the liver's basically losing the ability to maintain oxygen and so forth? And people argue about what hitting the wall in marathon running actually means. A lot of thinking is that it's actually resulting from the depletion of your stores of glycogen, which your liver is storing. So in addition to making glucose by gluconeogenesis, the liver can also release glucose, as she was referring to up here, by breaking down glycogen. And so it's thought that hitting the wall occurs when you really don't have hardly any glycogen left. So, but again, that's argued. Okay, other questions about that? Connie?
Yeah, speaking of hitting the wall, I've heard that after you hit the wall, you start burning up fat. How does that work? Because you said earlier.
Okay, so she says she's heard that after you hit the wall, you start burning up fat, and yes, you will be burning fat. You'll be burning fat to some extent as you're running as well. It's just that it's not as readily available source of energy for quick things at that time. But yeah, if you didn't have some backup energy source at that point, you'd be in pretty deep trouble. So burning up fat's important. Turns out burning up fat is very important for your heart. Your heart uses fat as a big energy source. And though I don't talk about it much here, another thing that your heart can actually use as an energy source is lactate. And so the heart can pull lactate out, convert it to pyruvate, but then instead of making glucose, it'll convert pyruvate to acetyl-CoA, again because the heart has plenty of oxygen, and acetyl-CoA is a source of ATP. So lactate can be used by the heart as a way of keeping the heart going. Of course, that's a pretty important thing to do, too. Other comments, questions? What?
I said generally.
Generally. It's generally considered an important thing. It depends on how well you like your relative, I guess. So, did you have a question?
Yeah, if the heart muscle has developed the ability to use lactate, why haven't regular muscles? The evolutionary wouldn't that be quite helpful?
Why haven't regular muscles developed that ability? Well, they would, but remember the regular muscles are away from the lungs. So they run out of oxygen. They could if they had oxygen, but they don't have that. In other words, if they had oxygen, they wouldn't be making lactate in the first place, right? So they're only making lactate because they have to when they're out of oxygen. So it's the oxygen that's determining that. It's not any limitation that's there of theirs. Make sense? How about a last song? The last song, I always get out of breath, so that's why I didn't sing it after the last one, because the last one is a long song. It's my favorite song I've ever written, so I hope you guys like it. It's to the tune of Supercalifragilisticexpialidocious. You know what it is. All right, here we go. It's called gluconeogenesis. When cells have lots of ATP and NADH2, they strive to store this energy as sugar, yes they do. Inside of mitochondria, they start with pyruvate, carboxylating it to make oxaloacetate. Oh, gluconeogenesis is so exhilarating. Memorizing it can really be exasperating. Liver cells require it, so there's no need for debating. Gluconeogenesis is so exhilarating. Glucose, glucose, come to be. Glucose, glucose, come to be. Oxaloacetate has got to turn to PEP, employing energy that comes from making GTP. From there it goes to make a couple phosphoglycerates, exploiting enolase and mutase catalytic traits. Oh, gluconeogenesis is liver specialty, producing sugar for the body most admirably. 6 ATPs per glucose is the needed energy. Gluconeogenesis is liver specialty. Oh, glucose, glucose, joy to me. Glucose, glucose, joy to me. Converting phosphoglycerate to 1,3-BPG requires a phosphate that includes ATP energy. Reduction with the electrons gives us all an NAD. And G3Ps isomerize to make DHAP. Oh, gluconeogenesis is anabolic bliss, reversing 7 mechanisms of glycolysis. To do well on the final, students have to learn all this. Gluconeogenesis is anabolic bliss. Oh, glucose, glucose factory, glucose, glucose factory. The aldolase reaction puts together pieces so a fructose molecule is made with 2 phosphates in tow. And one of these gets cleaved off by a fructose phosphatase. Unless F26BP's acting, blocking pathways. Oh, gluconeogenesis, a pathway to revere that makes a ton of glucose when it kicks into high gear. A cell's a masterminding metabolic engineer. Gluconeogenesis, a pathway to revere. Oh, glucose, glucose, jubilee. Glucose, glucose, jubilee. From F6P to G6P, that is the final phase. The enzyme catalyzing it is an isomerase. Then G6P drops phosphate and a glucose it becomes inside the tiny endoplasmic reticulums. Oh, gluconeogenesis is not so very hard. I know that on the final we will not be caught off guard because our professor lets us use a filled out index card. Gluconeogenesis is not so very hard. Yeah! Woo! Thank you. Happy Turkey Day. Hi, Kai. This is Kevin calling. Don asked me to let him know if we had any problems with the microphone in Milam, and it worked just fine, so no problems. Okay, thanks. Bye.
Microphone work?
Yeah, he checked it. There was a battery problem. So you got to change it for me. He actually, Don may come back and show you, but he put a battery tester in here, that if you have a question, you can actually pop a battery on there and test to see if it works or not. So if there was a bad battery in it is what it was. So.
Do you want me to ping Oscar when I'm
Hi.
Any movie ideas? Do you have a favorite Woody Allen movie?
My favorite Woody Allen movie? Wow. I love Bananas. One of his earlier ones. Why?
I just needed movie ideas for the break, so
For what?
I thought you were a fan because you had your poster in your office.
Oh, yeah, yeah. Yeah. Oh, I'm sorry. Yeah. I've got a one-track mind, Andy. I can't do two things. I'm not very good at multitasking.
How do you get out?
So to get out of this, you just have to log out.
Oh,
So,
okay.
yeah, sorry about that.
That's no problem.
Hi, how you doing?
Good, how are you?
Ready for